Welcome, my dear students, to this section on further on water-soluble vitamins. So, in this section, I'll be talking to you about folic acid. Folic acid, from where did the word come? It comes from a Latin word, folium. Folium means leaf. So, this is mainly present in green leafy vegetables, and hence it is called as folic acid, got from green leaves, and that is why folic acid. Now, what is the chemistry of it? Folic acid is having a pteridine ring, a papa plus glutamic acid. It is called as Pteroyl glutamic acid. Now, the glutamic acid imparts negative charges, so it cannot traverse membranes by diffusion because of the negative charge. And polyglutamation helps in the sequestration of folate in cells wherever it is required. Now, what are the sources? Minute you say folic acid, the word that should come to your mind is green leafy vegetables. So spinach, green leafy vegetables, fortified breakfast cereals nowadays contain folic acid. Animal sources, liver, yeast, etc. So many meats contain folic acid. Now what will happen is, important thing is food processing and preparation, because it can destroy up to nearly 50 to 90% of the folate in the food can be destroyed by just because of the processing. So raw foods are supposed to contain more amounts of folic acid. RDA for adult males is 100 micrograms per day. In pregnancy, it is 400 micrograms. Very important, folic acid in pregnancy. During lactation, it again decreases to 280 micrograms. Children, 25 to 200 micrograms per day. Now, what is important is pregnancy, because folic acid deficiencies are associated with neural tube defects, and nowadays it is called as folic acid supplementation should be given in the periconceptual period. Before the conception, during conception or after conception, and even at the time of conception, even in the first trimester of pregnancy, folic acid supplements are advised. So the requirement is 400 micrograms per day. Now, absorption. In the diet, folic acid is present in the form of polyglutamates. Now, in the intestine, there are what are enzymes called as glutamyl hydrolases. They all cut down this polyglutamates, release the glutamic acid to form monoglutamates or diglutamates. Now, then this can be absorbed. This folic acid can now be absorbed, either the monoglutamate form or the diglutamate form. Monoglutamate form is what is preferred. The folic acid, what we call as folic acid, is the monoglutamate form. This is absorbed and then it is transported bound to a beta globulin. Now, how is it synthesized? What is the active form and how is it synthesized? Now, folic acid, Pteroyl glutamic acid, is converted to dihydrofolic acid, DHFA or DHF. Some people write it as DHF, dihydrofolate. If A is written, it stands for dihydrofolic acid. And for this, the enzyme is folate reductase, which requires NADPH and vitamin C. So, vitamin C, one function is its action requirement for folic acid and synthesis of the active form of folic acid, vitamin C function. Again, in the further one more step, dihydrofolic acid, DHF, is converted to tetrahydrofolic acid, THFA. Again, the enzyme is folate reductase or folic acid reductase, and NADPH is required, vitamin C is required. So the active form is DHFA or THF, tetrahydrofolic acid. The hydrogen atoms get attached to the Pteroyl ring, and this is known as tetrahydrofolic acid. Now, what are the biochemical functions of folic acid? It is mainly involved in 1-carbon metabolism. Folic acid is the vitamin of 1-carbon metabolism. So this one carbon, where can it be attached? It can be either attached to the nitrogen in the 5th position or nitrogen in the 10th position, or it can be both. So we call it as N5 or N10 tetrahydrofolic acid or N5 tetrahydrofolic acid with the appropriate carbon compound, or it could be N5, N10 also. Now, the 1-carbon units that can get attached to folic acid include formyl, imino, methyl, methylene, and methynyl. I've given the structures out here, but mainly this is important is the names. Formyl, formimino, methyl, methylene and methynyl. These are the one carbon compounds that are carried by folic acid. There may be sometimes hydroxymethyl also, especially in serine and glycine metabolism. Now, which are the one carbon donors? From where did form tetrahydrofolate get its one carbons? So it comes from histidine. Histidine is one of the most important amino acid. Histidine is converted to urocanic acid. Urocanic acid is further converted to form-iminoglutamic acid, FIGLU, what we call as form-iminoglutamic acid. Form-iminoglutamic acid gives its form-imino group to tetrahydrofolate to convert it to form-iminotetrahydrofolate. So, histidine important is converted to FIGLU, and this is an important reaction. In this gives it to tetrahydrofolate and converts it into iminotetrahydrofolate. So that is the importance of histidine. So it has formed tetrahydrofolate has taken form-imino group from histidine. Which are the other one carbon donors? Now, other one carbon donors, one important donor is methionine. Methionine, remember how it acts as a one carbon donor, but mainly it is getting converted to S-adenosyl methionine, and there it is involved in methyl transferases. But for the reformation of methionine, it requires folic acid. So again, folic acid is methyl tetrahydrofolate is converted to folic acid, tetrahydrofolate. So somehow or the other it is linked with folic acid. Methionine, methionine. After that we have got choline, betaine, all these are involved in one carbon metabolism. Glycine is also there, serine is also there, all of which serine hydroxymethyl transferase is the enzyme which converts serine and gives the hydroxymethyl group to tetrahydrofolate. So these are the one carbon donors: histidine, methionine, glycine, serine. So we go ahead to, okay, it has accepted one carbon from all these compounds. What will the folic acid do with it? What is it going to do? So it is going to donate somewhere where it is acting as a coenzyme. Remember, everywhere it is the coenzyme for certain enzyme, and it is taking up all this one carbon from different compounds and it is going to give it to different compounds. That is its function, one carbon metabolism. Take one carbon from one place and give it to another. So the one carbon acceptors. Now one most important thing is it is involved in purine ring formation. Please remember the C2 and C8 carbon at the second position and carbon at the 8th position of the purine ring comes from folic acid, tetrahydrofolate. N-formyl methionine. So in this also the formyl group to the methionine has been donated by folic acid. So in this N-formyl methionine, what is it? Is formyl methionine of the tRNA. So tRNA which requires the formation of N-formyl methionine, it comes from folic acid. Homocysteine to form methionine, it acts as a donor. Ethanolamine to form choline. Choline is important in forming acetylcholine. So ethanolamine to form choline, again one carbon donor is going to be tetrahydrofolate. Uracil, one very important reaction is uracil is converted to DTMP. We saw how UMP is converted to UDP. UDP is forming deoxy-UDP. Deoxy-UDP is then converted to deoxy-UMP, which is then converted to deoxy-TMP. Whatever it is, from uracil, if you want to synthesize thymine, which is required for DNA, deoxythymidine monophosphate, then one carbon for tetrahydrofolate is essential. So from uracil to form DTMP synthesis, one carbon tetrahydrofolate is required. So these are the one carbon acceptors. Now just to give a good representation of the different interforms which are interchangeable among themselves. So let us look at this diagram. Dietary folic acid is there, as I said, and that is converted to dihydrofolate, folate reductase converted to tetrahydrofolate, folate reductase, and vitamin C. Then what happens? NADPH is also required for this. Then what happens? This tetrahydrofolate can also be got from N5-methyl tetrahydrofolate. Now during this conversion, it is vitamin B12 and methyl transferase, and homocysteine is converted to methionine. What else is there? There is histidine, which forms Fig-Glu. Fig-Glu converts tetrahydrofolate to formyl tetrahydrofolate and gets converted to glutamate. And then tetrahydrofolate can form N5-N10 methylene tetrahydrofolate by the reaction with serine and glycine. And this is converted to dihydrofolate. Remember, the difference here is usually it is reconversion from formyl to tetrahydrofolate, methylene to tetrahydrofolate. It is just tetrahydrofolate only what is found, unless in this reaction when DUMP is converted to TMP, it is dihydrofolate that is formed, and this TMP is going to go into DNA synthesis. So this gives an overall overview of the different interconversions in the tetrahydrofolate metabolism. So let us now look at the deficiency disorder. What will happen if folic acid is not there? So what are the causes for deficiency? It could be because of deficient intake. Intake itself is less. Pregnancy is one more reason where demand is more and the amount that is supplied is less. Defective absorption, very common cause, could be because of intestinal sprue, diarrhea, etc., wherein there is not enough time for the glutamyl hydrolases to act and to release the monoglutamate form to be absorbed. So again, defective absorption. Now there are certain drugs like hydantoin and phenytoin. Again, these also prevent the conversion of the polyglutamate form to monoglutamate form and thereby inhibit the absorption of folic acid and lead to folic acid deficiency. Hemolytic anemias, the demand is more because in these cases what is happening? The hemolytic anemia, RBC's bone marrow is keeping on dividing, and every time it is dividing, the DNA replication is taking place. DNA replication requires TMP, and for that to be synthesized, the only and only route is through folic acid. Since folic acid, if it is deficient, everything will be affected. So hemolytic anemias, again the demand is more and it can lead to decreased folic acid. Now there is what is called as folate trap. Because of this, I'll talk about this along with B12. Now deficiency manifestations, there will be reduced DNA synthesis. Basically it is all because of DNA, and it's the most important reaction in which DUMP is converted to TMP, folic acid is required. No TMP, no DNA synthesis. So reduced DNA synthesis. And which are the cells which are affected? Bone marrow, intestinal cell. They are the ones which are most affected. Why? Those are the cells which are rapidly dividing. Minute a cell is rapidly dividing, it will require replication. Replication means DNA synthesis. So other deficiency manifestation will be because of all these effects only. It can result in macrocytic anemia. Now here there is bone marrow is affected, so there is a mismatch between DNA and protein synthesis. They won't go hand in hand. So hemoglobin accumulates in the precursors and it will result with having immature nucleus with a mature cytoplasm. The WBCs are also affected, can lead to leukopenia. There will be also hyperhomocysteinemia. Remember, N5-methyl tetrahydrofolate is required to convert homocysteine to methionine. If that does not happen, it can lead to hyperhomocysteinemia. Now, one classical thing that is seen with folic acid deficiency is spina bifida, birth defects. The mother should take folic acid supplements, otherwise it can lead to birth defects including like spina bifida, and it is can also be implicated in cancer. Supposed to prevent cancer, extra doses of folic acid. How do we assess folate deficiency? One is by the serum levels, the other is what is called as the histidine load test or the Fig-Glu excretion. Again, in this what is happening, we give a load of histidine and we check for the excretion of Fig-Glu. If this is not there, if folic acid deficiency is there, Fig-Glu excretion will be more. So this is known as the histidine load test, and peripheral smear definitely helps to know about the different types of anemia that is seen in folic acid deficiency. We'll go ahead with the next vitamin, vitamin B12. Now this is also called as cobalamin or anti-pernicious anemia vitamin or the extrinsic factor of Castle. What's the chemistry? Contains one cobaltamine. So that is the most important thing about B12. It contains cobalt. It has 4 pyrrole rings, 1 benzene imidazole ring and a R group. So structure schematic diagram looks like this. Now what is the different forms? Now if the side group is cyanide, you call it cyanocobalamin, which is the oral form. Hydroxycobalamin will contain the OH group. It is the injectable form. Adenosylcobalamin will contain adenosine, and the 5'-deoxyadenosine is the storage form and the coenzyme form. And if it contains the methyl group, you call it the methylcobalamin, and the coenzyme form and seen in circulation and cytoplasm. It all depends upon this R group. Now let us go to the sources. What are the sources of vitamin B12? B12 does not have any plant source. They are all foods of animal origin. So what are the foods? So vegetarians, can they develop B12 deficiency? Yes, maybe there are chances of developing B12, but most of the vegetarians drink milk, and milk is an animal source, and thereby they may not develop because of the milk and milk product consumption. But it is present in liver, kidney, eggs, fish, pork, chicken, etc. Milk and curds for the other people, and plus there is supposed to be some amount of intestinal bacteria do synthesize B12 by the intestinal bacteria, but however its absorption is questionable from the intestinal bacteria, because where it is taking place, synthesis is taking place somewhere else and absorption is taking place somewhere else. Now RDA is 3 micrograms to all these other different RDAs in different situations. Now let us go to the absorption. Now protein-bound cobalamin is released by acid hydrolysis present in the stomach, and cobalamin binds with the intrinsic factor of Castle, IF. IF is a glycoprotein, intrinsic factor secreted by the parietal cell and combined with 2 B12 molecules. This prevents the degradation of cobalamin, and the absorption takes place in the ileum. It prevents and it carries it B12 along with it, and in the intestinal cell it is going to get absorbed in the intestinal mucosal cell. Now the transport methyl, it is mainly methylcobalamin, which is transported with the help of transcobalamin 1 and 2. In the liver, it is stored as adenosyl B12 combined with the protein transcorin. So transcobalamin and transcorin are required for the further transport and storage of vitamin B12. What are the functions of B12? Only 2 enzymes within the body require cobalamin as coenzyme. In one, it will be adenosyl cobalamin, which is acting as the coenzyme, and in the other, it is methylcobalamin, which is acting as the coenzyme. So the 2 enzymes are methyl malonyl CoA mutase and homocysteine methyl transferase. Let us just see where it is involved. Now all these metabolism, propionyl CoA to succinyl CoA. So this enzyme L-methyl malonyl CoA mutase requires adenosyl B12 as the coenzyme. Now homocysteine methyl transferase. Homocysteine is converted to methionine. In this, methylcobalamin is involved, and it is regenerated by taking a methyl group from methyl tetrahydrofolate. So this is the connection between B12 and folic. What will happen if B12 is not there? This methyl tetrahydrofolate will remain like this. There is no other reaction in the body where this methyl tetrahydrofolate can be converted to tetrahydrofolate. So what will happen if B12 is deficient? Folic acid deficiency also arises. Why? THFA has to have N5N10, methylene tetrahydrofolate. Only then it can convert the DAUMP to TMP. So all the deficiency manifestations of folic acid also develop when B12 is deficient. This is called as folate trap. The only way free tetrahydrofolate is generated is by this type, and if B12 is deficient, folic acid is trapped as methyl tetrahydrofolate. No THF to do other functions. So this is known as methyl folate trap or folate trap. So in B12 we get folic acid deficiency also. So let us look at the deficiency of cobalamin. What will happen? What are the causes? It may be nutritional, maybe because of low socioeconomic or vegans, decrease absorption, why intrinsic factor is required. So gastrectomy, ileum resection, then the absorption will be affected. There is a condition called as Addisonian pernicious anemia, and gastric atrophy, pregnancy, etc. may be involved in deficiency of cobalamin. Fish tapeworm is also implicated. What are the deficiency manifestation? Megaloblastic anemia, methylmalonic aciduria, abnormal homocysteine level, homocysteinuria, demyelination, subacute combined degeneration, and folate trap and deficiency manifestations of folic acid. So what is happening? Higher homocysteine levels are linked with myocardial infarction. Active methionine is required for the methylation of phosphatidyl ethanolamine and choline, and that will deficiency. These are phospholipids. So myelin sheath is affected because of the deficiency of these phospholipids. That's why we have the leads to demyelination. B12 deficiency leads to demyelination. Now one word about pernicious anemia. Pernicious anemia is an autoimmune disorder due to antibodies against the intrinsic factor and parietal cell. It is a type of megaloblastic anemia. So anemia is accompanied with neurological manifestation, subacute combined degeneration of the spinal cord, associated with all these symptoms including absence of HCl. And with this we come to the end of vitamin B12 and folic acid and pernicious anemia. How these both are related. Folic acid, B12 is deficient, it will also lead to folic acid deficiency.